everyone, and welcome to today's webinar, Exploring the Pillar Biosciences Research Use Only, RUO, oncoReveal Solid Tumor 22 Gene Panel Kit, and Biomech NGenius Next Generation Library Prep System. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit www.beckman.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I do also want to mention that there is a survey at the end of the webinar. If you could please take a moment and submit your responses when prompted to do so, that would be greatly appreciated. I'd like to now welcome our speakers, Dr. Nicholas Lodato, Principal Scientist, Research and Development with Pillar Biosciences, and Dr. Enrique Neumann, Global Marketing Development Manager at Beckman Coulter Life Sciences. Dr. Lodato, you may now begin your portion of the presentation.
Great, thank you. So before we get started into some of the really cool work that we've been doing with Beckman, I want to tell everybody a little bit about Pillar Biosciences, who we are and what we do and what motivates us. So despite NGS being a technology that's been around for a while, clinicians and patients often struggle with access to this platform. This is because of a few reasons. So for one, NGS testing is typically done at large central labs, so the clinicians have to send their patient samples out and wait to get data, which obviously leads to delays. And two, sending samples out to an external lab is often very costly. So at Pillar, we aim to solve this, or both of these problems by decentralizing NGS testing. We do this by manufacturing distributed kits and a kit-based approach, so allowing testing to occur oftentimes in the same hospital that the patient's biopsy is taken. And so, ultimately our goal is to get the patient the right therapy faster. And so to do this, we combined several proprietary technologies with a broad portfolio. So on the technology side, our library prep is highly accurate and it's amplicon-based, so it's very reliable. It's a simple workflow, so one tube, even if you have overlapping regions of interest, one tube is all that's needed. And so the library prep itself, too, is also designed and developed with automation in mind, so it's a very automatable workflow. The workflow is very efficient, so you could prepare libraries and have it loaded on the sequencer within 1 day, and then with sequencing time, within 2 days, you get the patient's data as opposed to a few weeks sending the patient data, or sending the patient sample out, rather. So our portfolio, so we currently have over 20 oncology-based RUO products, but we also have a few that are approved by regulatory bodies. So the FDA, we have a product that's CE marked, and also a approved product in China by the NMPA. We also have CLIA labs that we work with. So we have one here at Pillar in the US that is mainly focused on the pharma partnerships, and we also have one in China that does a lot of the clinical testing. So these are sort of the three pillars of Pillar, as we like to say. So the first is versatile. So if either a customer or if we want to develop a new panel, we come up with a bunch of gene targets and mutation targets. Versatile is an AI-enabled automated software which can do all the primer design with a very high capability. So we oftentimes can design hundreds of different primers that can amplify all in one tube, and this is all done through the design in Versatile. The second is the library prep chemistry itself, so using our SlimAmp enrichment chemistry, which I'll talk about in a few slides. So this technology, again, it's all done in a single tube, even if you have very complex regions of interest. It's a very fast workflow, as we'll get into later as well. Again, fully automatable. It was developed with automation in mind, although most of the testing to date before this partnership with Beckman has been just doing all manual preps. It works well on very damaged FFPE samples. We've heard this from customers. So FFPE samples that fail other assays work really well with our SlimAmp technology. The third pillar is the bioinformatics platform PIVAT, which is something else that we've developed here at Pillar. This is software that we like to say is a one-button sample to report. So it takes the raw output from a sequencer, and just with a few clicks, you get the patient data in as little as a few hours. So our portfolio, as I mentioned, is over 20 commercial products strong at this point, including many solid tumor panels. So the oncoReveal 22 gene solid tumor panel, highlighted in green, is the one that we'll be talking about today. But we also have a few more panels in mind for further development. So in terms of solid tumor, not only do we target SNPs and SNVs in our solid tumor panels, we also have an MSI panel, which can call MSIs, as well as some fusion panels as well. We also have some solid tumor panels that are designed for liquid biopsy approaches. So that's our oncoReveal Core liquid biopsy, as well as some other panels as well. So for blood tumors, we have a myeloid panel, which targets a lot of really important variants within myeloid diseases. And recently, we've been working with the BRCA and HRD type of diseases. So we, of course, have BRCA1 and BRCA2 somatic and FFPE, SNV, and indel detections, but we also have been doing a lot of really great work developing panels for methylation testing. To talk a little bit more about SlimAmp and how it works. So with conventional PCR, if you have two regions of interest that are overlapping, if you were to simply design two sets of primers, of course you will get those regions of interest to amplify. But the problem is if you notice the reverse amplicon or the reverse primer from the amplicon-1 and the forward primer from amplicon-2 are facing each other in this scenario, right? And this produces a inner amplicon, which is much smaller than the two regions of interest. And given that PCR is largely, or PCR efficiency rather, is largely dependent on the size of the amplicon that you're trying to amplify, smaller regions will take over a reaction if given enough cycles, right? So after n number of cycles, you'll get some of your region of interest to amplify, of course, but most of the product that gets formed is actually the small inner amplicon. And obviously in this scenario, if you were trying to sequence this, you would get a lot of unwanted reads from that inner amplicon. But with SlimAmp, again, taking those same two regions of interest, so we have a patent on the ability to take a complementary sequence from the forward primer of amplicon-2 and add it to the reverse primer of amplicon-1. And then once that inner amplicon is formed, those two complementary sequences bind to each other, forming a stem loop. This stem loop is not amplified during subsequent rounds of PCR. So while, given n number of cycles of PCR, while you get exponential amplification of your two ROIs, the inner amplicon is only amplified in a linear way at best. So, doing enough amplicon or doing enough PCR cycles, you could effectively dilute the inner amplicon to be near zero. So to compare ourselves to other conventional workflows, NGS workflows. So with conventional workflow, there are often several steps, including ligations and multiple amplification steps and indexing with cleanups in between. And because of that, because of the multiple steps, this often, has to take place over a course of 2 days or, 10.5 hours total. But with our oncoReveal workflow, it's much more streamlined. So instead of having ligation steps and extra steps, we simply have 2 PCR steps and 2 cleanup steps, and that's it. And then, because we have fewer steps and the flow is actually optimized to be very streamlined, all of this can be done within 6.5 hours, going from extracted DNA to something that's ready to be sequenced. Additionally, there are multiple stopping points. So after each PCR step and after each cleanup step, the technician or whoever's preparing the libraries is more than welcome to put the plate in the freezer and come back the next day. So it's very flexible in that way. So to sort of recap the SlimAmp technology, so we could amplify many regions at high plex rate in a single tube, including those with overlapping ROIs, and that reduces the need for pooling after. So it requires fewer reagents to actually get your sample from your sample to a prepared library. It's a very simple workflow, 6.5 hours with multiple stopping points. And again, automation was always sort of like in our mind when we were developing this technology. And something I don't have time to talk about today, but PyVat combined with our chemistry, we have low failure rates, and we were able to detect variants at very low frequency without the use of UMIs. So now to get into sort of what we've been doing with Beckman Coulter. So we decided to go with our oncoReveal 22 gene solid tumor panel as the first panel to automate on their platform. This is a panel that's actually an RUO version of our FDA-approved oncoReveal CDX panel. And again, everything I'll be talking about today was using the RUO reagents and RUO PyVat. This assay in particular was chosen because it has a lot of key mutations in a lot of key mutations in many solid tumors, including non-small cell lung cancer, colorectal cancer, melanoma, and bladder. To go into a little bit more of what variants that we cover within this panel, on the top here, these are the 22 genes that are covered. So ALK, BRAF, DDR2, et cetera. And then to go into a little bit more detail, these are the sort of key mutations that are found within these highlighted genes. So for EGFR, the drug targets L858R and exon 19 and frame deletions are covered. For KRAS, G12 and G13, of course, are covered, as well as many others. EGFR T790M, the drug resistance mutation, that's covered. And again, this is just a sampling of what's there. This panel covers many thousands of variants. So the Pillar workflow, so 2 days total, including one day of library prep, overnight sequencing, and data analysis. So for the library prep, it takes about 3.5 hours of hands-on time if you're doing a manual process, and then again 6.5 hours of total time. So if all of those, if the libraries were prepared in one day and then sequenced overnight, the next morning the researchers can load that data directly into PyVat. PyVat takes about 2 hours to analyze a typical MiSeq or NextSeq run, and then a few hours of tertiary reporting. So you can see here within 2 days, certainly within 2 days, you could go from extracted DNA to an actual patient report. Where our partnership with Beckman comes in is we could actually reduce the amount of hands-on time that's required for the library prep from 3 hours to as little as 30 minutes. The total time stays relatively the same. A lot of the time is actually in the thermocycler or just manipulating the samples, but the hands-on time is really important. So to go from 3.5 hours of hands-on time to 30 minutes is pretty big. And a lot of this, and all of this 30 minutes actually is all up front. So all of this is just loading the samples, loading the reagents into the machine, and then the rest of the 6 hours is sort of walk away time. To go into detail of what the Ingenious system is doing on board. So all of the steps in blue are on board. It takes the DNA sample and the reagents and it prepares the plate for PCR. It has an onboard thermocycler, so it does all of the cycling on board. Then it has the ability to do the PCR cleanup. So after gene-specific PCR, the first PCR cleanup. And then it prepares the second round of PCR, which adds the Illumina indexing adapters. Again, the cleanup is very similar to the first cleanup, so it of course does that. And then it finally outputs a final library in a plate. The only thing somebody would have to do manually at that point is to just do some simple QCs. So the library quantification and then pooling and obviously loading into the sequencer. So to compare the manual prep to automation, so for manual prep, we say you could batch up to 24 samples. For the Ingenious system, it's between 4 and 24. The input is the same, so between 10 and 80 nanograms of DNA, and both platforms support either the use of genomic DNA or FFPE. Again, the biggest difference between the two is actually the amount of hands-on time that's required. So for manual prep, between 3 and 4 hours is pretty typical, depending on the number of samples. But then with the Ingenious system, it's as little as a half hour. And that difference is mostly made up in the amount of hands-off time, right? So for manual prep, 3 hours of hands-off time versus 5 or 6 hours plus of hands-off time for the Ingenious. So to do a head-to-head evaluation, this is the experimental design that we came up with. So because this assay can support genomic DNA and FFPE, we chose one of each. So for genomic DNA, we used the genome in the bottle cell line DNA NA12878. The input for that was 10 nanograms. For FFPE, we used the Horizon Moderate formalin compromised DNA standard, which is at 20 nanograms. Total libraries per run for Ingenious, it's a 24-well plate system, so we chose to either do 1 column of 8, 2 columns for 16, or the full plate for 24. And for manual prep, we just did a single column of 8 samples. For total runs, we did 3 Beckman Coulter runs, 1 of each of the samples, so 1, 8, 1, 16, 1, 24. And for manual comparison, we did 2. So all the libraries were sequenced across 3 Illumina NextSeq DX runs using mid-output flow cells. And this is honestly mostly because we combined them just internally with our own R&D other runs. All of these libraries can more than fit on a NextSeq or a MySeq flow cell as well. The secondary analysis and variant calling was all done by the PyVat bioinformatics software. So to get into the comparisons between the two and how they both performed. So on the left here, this is the pre-sequencing QCs. This is the final library yield. The yield is in nanomolar. So in black is the manual, in blue is the Beckman Ingenious platform, and then NA12878 and moderate formalin compromised DNA. You can see here that the Beckman platform actually performed a little bit better than the manual prep. Although this 3.5 nanomolar line here, this is what we would consider like a good library prep. So anything above that is good. But you know, the Beckman Coulter system did outperform the manual prep here. And then after sequencing, these are some QC metrics that we like to look at. So the mapping rate and the on-target rate. On-target meaning the targets within the actual 22 gene panel. And so for the manual prep, the mapping rate was, they were both at 100%. Ingenious, it was just very slightly below that, like 99.5 or 99%. On target rate, again, I drew this line here at 90 because we consider anything above 90% on target to be a pretty good sequencing run. And although there are some small differences between the two platforms, all of the libraries had very strong on-target rates, around 95+%. So you know, taken together, the Beckman Ingenious platform produced very good, high-quality libraries with very good mapping and on-target rates. And then, you know, maybe one of the more important things is how well do the libraries actually capture the variants that are within the sample. So here we're comparing the manual variant allele frequency that was detected by PyVat on the x-axis versus the Ingenious on the y. And these are all the variants that were in the Horizon Moderate FFPE sample. And you can see that there's a very, very strong agreement between the two platforms. So slope of 0.99, R squared of 0.99. Throughout the entire sort of like spectrum of variant allele frequencies that are in that sample, so anywhere from 25% all the way down to about 2%. And in particular, what was impressive to me was the 3 variants here that are all below 4%. So they're all EGFR variants, the L858R, the exon 19 deletion, and the T790M. All 3 of these variants were in very strong agreement between the two platforms. So in conclusion, using the Biomech Ingenious system with our assay, 24 libraries can be prepared in approximately 7 hours with about 1 hour of hands-on time and 6 hours of hands-off time. The automated platform can produce really high-quality libraries using the 22-gene solid tumor panel. And the resulting variant calls for libraries prepared on the Biomech Ingenious system were as expected and comparable to manual prep. And then so some future work, as I mentioned before, we plan on trying to or putting some more of our panels on the automation platform in the near future. So the myeloid panel is certainly one that we are aiming to develop next, as well as another version of a solid tumor panel, a multi-cancer panel that has CNV detection capabilities, as well as a liquid biopsy panel in our core LBX panel. And with that, I'll pass it over to Enrique. Thank you.
Hello, everyone. So thanks a lot, Nicholas, for presenting the very good results and the very good performance of your reagents on our instrument. And now that we've seen how that performs, I'm going to talk to you and I'm going to show to you a bit more about the instrument itself and explain you how it works. First of all, for the ones that might not know us, so Beckman Coulter Life Sciences, we're one of the biggest providers in life science instrumentation globally. And our mission is really to empower those seeking answers to life's important scientific questions and healthcare questions. And I think that resonates very well with what I'm going to show you in my next slide. That it's a bit how we started designing and what was the thought behind the ingenious. So actually what we say is that for the design of this instrument, customers spoke and we listened. So these were things that we were hearing from genomics and specifically NGS customers. For example, that human-associated errors can cost a lot of money, given that reagents are usually costly, and any human error made can actually make you waste all those reagents as well. That automating workflows, the need to automate workflows, it's because in labs people don't have the time, the hands-on time to work, and they want to work with a system that it's easy to work with, that doesn't need any type of babysitting. As well, something very common with this type of customers is that they don't have necessarily programming skills, and they don't have the time to actually learn how to program an instrument. So they needed something very easy to set up and very easy to run. And finally, the need of flexibility. So NGS, it's a very dynamic type of technique. So they need to make sure that the instrument they have is adaptable to new technologies that might be coming. So for this, we came up with the ingenious. So the ingenious, it's a library preparation robot. Very compact, and actually the main features of this instrument is that it's very easy to automate a very error-prone process as is NGS library prep. It's very easy to set up applications. It really reduces hands-on time. And the greatest thing is that with the same instrument, with the same configuration, you can run a very wide array of kits of multiple chemistry vendors. So let's go bit by bit into the different characteristics of this system. And I think this is one of the greatest ones because it's very common when we're setting up a run on a robot to actually make mistakes, to place something where it shouldn't be, or, for example, to have a plate with a lid where we shouldn't have a lid. And that can actually very easily go downhill, and it can make us lose the whole run that we are performing. So for this, we have this solution, the dynamic deck optics system. So up in the roof of the instrument, let me see if I can get a pointer. So up here, there is a camera. And that camera, it's actually scanning live what is happening on our work table. So the instrument will be confirming real time as we load things if we are loading them in the correct position. And as well, it's able to recognize, for example, if we are loading vials that shouldn't be capped and they are capped, or if there's a lid or not in one of the reaction vessels. So this is actually a very, very valuable tool to make sure that when we kick off a run, we make sure that what we have on the work deck, it's ready to go and perfectly placed. As well, we have a system that is able to identify liquid level. So reagents will go more into detail of this carousel in a moment, but actually we are loading reagents as they come in the kit into this carousel. This carousel goes into the instrument, and we have a liquid level detection system that is able to calculate the amount of liquid on each of the tubes, and it will let us know if we have enough volume for the run that we want to perform or not. As well, there is a second camera here that it's going to be scanning all those tubes, and it's going to read through the labels of those tubes so that way we can make sure that we have the right reagents in the right place. So this input carousel, the main advantage of this is that it really makes the whole process a lot easier, a lot more simple, because we don't have to do any pipetting beforehand. We don't have to prepare any master mix. The instrument is going to prepare that for us when we start the run. So the carousel, it's going to be able to rotate, so all the tubes are scanned. Each position rotates as well, so it can read through all those labels. And all these features make it very, very error-proof, and it really reduces hands-on time. One thing that I want to mention about this carousel is that it's very easy to place on and off. So actually one of the features that we have is that we're going to place the tubes as they come from the kit into the carousel. The instrument is going to move those reagents, the amount that is going to be necessary for the run, to cooled positions. And then we are going to be able to retrieve that carousel and put those tubes of our valuable reagents back into the freezer or fridge for optimal conservation. As I said, we do have cooling positions. So we have 4 positions that actually span from 2 degrees up to 65 degrees Celsius. So that way we can keep our reagents in the best conditions for the moment they're going to be used, and as well we can heat up reagents if necessary. As well, to have even more hands-on, to reduce the amount of hands-on time, we have an on-deck cycler. So the on-deck cycler that we're using, it has been purpose-built for this instrument and purpose-built for our reaction vessels that I'm going to show you in a few slides. And it's a cycler that really assures that we have the minimal evaporation possible. And at the end of our run, it can keep our libraries ready to be sequenced up to 16 hours at 4 degrees to make sure that we can leave the instrument running unattended and then come back to retrieve our libraries with the certainty that they have been kept in the best conditions and they are ready to go into a successful sequencing run. The pipetting head in this instrument, it's actually an 8-channel pipetting head that is compatible with 2 tip box formats. So we have high volume tips that we have 96 tips per box, and we have the low volume tips that come in a format of 384 tips per box. So that allows us to reduce the amount of space necessary for tip storage on the instrument, and as well, it allows us to do complete runs without having to keep feeding the instrument new tip boxes. This pipetting head is able to select any number of tips from 1 to 8. So it doesn't need to work in blocks of 8. It can work with just 1. And this allows us, for example, to do that aliquoting from the tubes to our reaction vessels. And as well, the pipetting range of this head goes from 1 up to 1,000 microliters, which really makes it compatible with virtually any NGS library prep kit out in the market. As well, this same head, it has on the sides an integrated gripper. So this gripper, it allows us to transport lids and to move reaction vessels across the deck. It gives us the freedom to have longer walk-away times because the instrument, thanks to this gripper, will be able to move, for example, our reaction plate, our reaction vessel from a magnet out of the magnet and back into the magnet again for the bead washes, for example. And as well, something that I really like about the way the instrument has been designed is that everything is actually thought through. And whenever something is moved on the deck, the instrument will follow a path to avoid as much as possible flying over positions that can be contaminated, to avoid any type of contamination when we're doing the library preparation. Something else that is very handy in this case is that the instrument is able to recognize partial tip boxes. So if we have a tip box that is partially used, we don't need to move those tips. We don't need to discard it. We can place it as it is on the instrument, and the instrument, thanks to that deck optics system, the dynamic deck optics system, it's going to be able to read through the tip boxes and know where to start. And as well, it's going to be able to count how many tips we have. It will know how many tips we need for the run that we want to perform. And if they're not enough, it's going to give us a warning, and it's going to tell us that we have to actually load a tip box that has more tips in it. I mentioned a few times this reaction vessel that we're using. So we're not using classic PCR plates. We're using reaction vessels that are for 24 samples on the instrument. That allows us to actually reduce the footprint necessary for the reactions. And as well, this multifunctional reaction vessel that has been designed on purpose for the Ingenious, it has some advantages because it has a much bigger capacity than classic PCR plates. So it allows us to do high volume cleanups without any problem, as each of those wells can accommodate up to 600 microliters. They are barcoded. We have up to 100 million unique barcodes to make sure that we enable maximum traceability. And it is compatible, of course, with the thermocycler that we have on deck. How it is to use the system? Very simple. As I said before, you need no programming skills to be able to run this instrument. And actually, the instrument itself, it's not even attached to a computer. So everything you need to do is you need to know how to use this navigation dial. So this reminds me a lot to the first generation of iPods that had this wheel and a bottom. It's actually the same logic. You go around, you select whatever you want to run, and it's very, very easy to navigate through. To set up these runs before they go into the instrument, we do that through the Biomech Genius portal software. So we can do this remotely from any computer. As long as we have internet, we're good. Where we set up the run that we want to do. So we define, for example, the sample number that we want to run, the parameters that are variables in the IFU of the kit will continue to be variables in our run. So we can select those, and once we're ready, we just upload it to the cloud, and that goes directly into the instrument where we can download it and start the run straight away. This protocol, this portal software, once we define the protocol that we want to run, we define that batch of samples, it will generate a work aid PDF that will list all the reagents and all the volumes that are necessary. So it makes really our life a lot easier if you want to prep those reagents beforehand and before going into the instrument itself. During the run, the instrument actually, it's very informative from far away. So there is a light that you see here in multiple colors, and depending on how that status light shows, it's going to give us information of what's happening. For example, if it's blue, the instrument is ready to run. If it's green, there's a running process. If it's scrolling rainbow, is that the run has been completed successfully. So we can see from across the room what's going on with the instrument. You're not in the same room, that's not a problem. You can see what's going on with the instrument as well. Because from the Ingenious portal software, the one I just showed you, you can see the status of your instrument remotely from anywhere you want. The sample number that we can process for any of the protocols on the Ingenious goes from 4 to 24 samples. And when I say 4 to 24, it's really 4 to 24. It's not that we have to run batches of 4, 8, 12. We can do 6, we can do 12, we can do 16, we can do 17 samples if we want. So we don't have to actually have a set number of samples to be able to start the run. As long as we are between those 4 and 24, we're good to go. And the software will automatically calculate reagent volumes, tips necessary, and everything. So that way, in a very seamless way, you can get your, you can start your run without having to do any type of calculation. As well, we have different levels of security on the instrument. So different users might have different permissions. So an administrator might be able to run several instruments and set up runs, and operators might be only able to start runs from the instrument. So we do have different levels of users in the system to make sure that we can keep each person doing, seeing what they should do, and if we need someone to, for example, validate the run, only that person will be able to have that type of permission. As well, it's an instrument that is very scalable. So administrators can actually have several instruments on their portal software. Those instruments, they don't need to be geographically in the same place. And this way, you can do a division of pre and post PCR labs, or you can have multiple lab locations in different geographies and still have one admin working from one location that can have an overview of what's going on with all those instruments. Adding most instruments to this portal, to this user, is very easy. As soon as you install the instrument, you get an activation code. You put that code into the portal, and your instrument is immediately connected to your user. Something that I think is very important from this instrument is that actually, as I said, there's no need to have any programming skills. And you can access any method that is on our application library. So we do have a list of methods that is here. We're working in new methods for DNA and RNA sequencing that will keep appearing. So that way, we make sure that the system is future-proof and that you can start using no matter which of these methods. You can adopt it very easily and very quickly without the need of having to program absolutely anything. So to end, I just want to open for questions.
Thank you, Dr. Lodato and Dr. Neumann, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the lower left corner of your screen, and we'll answer as many of your questions as we have time for. Let's go ahead and get started. Our first question here asks, what kind of controls you use per sample basis to assure the library prep is controlled from lab to lab?
So is this for me, for the library prep part? I would assume. So I mean, for us, yeah. So like a lot of our kits, you know, some of them come with controls. Some of them, you know, we just recommend what the controls for a user to use. And so, you know, it's sort of a mix. So again, like obviously it's very important to have controls, right? So that you know the library prep went well, so you could like correctly assess the samples themselves. And so, yeah, for FFPE panels, Horizon makes a lot of controls. And so it just depends on the panel, right?
Thank you. Our next question we have says, let's see, why the automation WF has higher yields when compared to manual?
Yeah, so that's a good question. You know, a lot of the times for library preps with manual pipetting, you could lose a lot of your product in the ampere cleanup step, especially if you're not careful with disturbing the beads and not having a good wash. There's a lot that can go wrong manually. So I'm guessing it's because the automation is maybe more precise. Maybe it's just better at recovering the actual product that's being formed. That's sort of my guess. But the manual prep also produces really plenty of material for sequencing. But yeah, that's sort of my guess.
Great. Thank you. All right. Our next question that we have here says, do you need one or two liquid handlers to separate the pre and post PCR processes?
I think it's for me. So actually, if you need to separate pre and post PCR, you do need to have two instruments. Reality is that we have a good amount of users that are running the whole process with the same instrument without any problem. The instrument has a UV light for decontamination. So if necessary, you can do between the post PCR and the next pre-PCR run a decontamination with the UV light.
Fantastic. Thank you. All right, our next question here asks, how much time did it take to develop the method for the robot to work efficiently to the end result, and how many iterations did it take?
Is that the method for the library prep? Is it?
I can repeat the question if you'd like.
Yeah, can you repeat it? Sorry.
Yeah. How much time did it take to develop the method for the robot to work efficiently to the end result, and how many iterations did it take?
Yeah, I mean, it took for us, because we were developing it sort of from scratch, right? So developing the workflow from the beginning. So it took probably a couple months. I mean, a lot of it was just working out exactly because the Ingenious is very flexible, right? Exactly where the things should go on the carousel and how to go about the actual workflow. But for this panel, it was actually not too bad.
Great, thank you. All right, our next question here that we have says, Pillar's technology and oncoReveal panel in combination with the Ingenious seems like a good solution for upfront NSCLC screening, where tissue may be limited and time is of the essence to identify driver mutations for targeted TKI therapy. Can you confirm?
Yeah, so we target specifically the EGFR mutations, the L858R and the exon 19 deletions. Because they do have targeted TKI therapies. Again, this version that we automate here is RUO. But again, the instrument can take the DNA, extract it all the way to reporting within 48 hours. So yeah, I mean, it's certainly a quicker turnaround than having to send it out. And then, with the automation, it's very little like hands-on time. So yeah.
Great, thank you. All right, our next question here. It says, it sounds like the combination of Pillar's panels and Ingenious could provide genomic data within 48 hours for downstream clinical reporting. Would this be true of the other applications that Pillar is developing, and can you elaborate?
Yeah, so the answer is pretty much yes. So, I mean, I think one of the coolest parts of our technology and our library preps is they're very similar between panel to panel, right? So if the target's changing, then it doesn't really affect the two PCR steps too much. And so from that point of view, yeah, any of the FFPE or genomic DNA panels can all be sort of done with almost the same workflow. So yeah, within 48 hours for almost any of our panels on this platform, for sure.
Fantastic, thank you. All right, our next question here that we have asks, for the instrument, do I need to program or create my own methods?
No, actually, it's all the methods are pre-programmed for you. It doesn't matter if you're changing the sample number, if you're changing the variables that are in the IFU of the kit. You don't need to do any type of programming whatsoever. So the instrument is quite dummy-proof and very, very easy to use.
Great, wonderful. All right, you guys are going rapid fire. I love it. Our next question here says, it sounds like Pillar's technology can provide data on very little sample input. Has there been any validation performed on these assays to highlight the low end of the input required for the assay?
Yes, so the other solid tumor panel that we want to put on the Ingenious platform, that one we can go down as low as 2.5 nanograms of input. And so with some other manual testing, we could have like a greater than 95% success rate detecting mutations.
Oh, fantastic.
With that low.
All right. Our next question here that we have says, just how easy is it to use the Biomech Ingenious system, and how much automation experience will my team need to use the instrument?
So automation experience, none. Really, you don't need to have absolutely any experience to be able to use it. And if you want to get an idea of how it is to run the instrument and how it is to work in the everyday, I think it's very convenient that we have this QR code here in the slides. You can scan it, and we have a virtual 3D lab in our website in which you can actually see how it is the day-to-day operation of the system. So I really invite you to go there, scan that code, and give it a try, because it's a very good tool to get an idea of how it is to work with it.
Great, thank you. Our next question here says, what is the minimum dead volume required?
The minimum dead volume, it's defined both by the instrument and by the type of container that we're using. And I would say that this is the dead volumes we have are comparables to the ones of any classic liquid handler. So it really depends as well on the viscosity of the liquid. So it's hard to give a certain number, but this is minimal to make sure that we are impacting the performance of your kit in sample number as minimum as possible.
Great, thank you. All right, and it looks like we have time for one more, and that question says, can it pause after a certain step?
Actually, yes. So that's one of the beauties of this system, is that you don't need to run your method from A to Z. You can actually run it partially. By running partially, it means that you can stop in any point that is considered a safe stopping point in the protocol. So, for example, if we have a protocol that it says after a certain bead cleanup, we can freeze those samples and continue later. That's not a problem with the Ingenious. You will be able to stop there and resume later on in time, the day after or the week after if you want, without any problem.
Thank you both. And I want to take this time to ask Dr. Lodato, do you have any final comments for our audience before we go?
No, I think I'd just say that, having our panel on the Ingenious really helps to just get libraries prepared faster, right, and more reliably. And so I think it's a great platform.
Fantastic, thank you. And Dr. Neumann, did you have any final comments for our audience?
No, really. What I want to tell you is that really this fantastic platform, very easy to use. And as I said, I really invite you to scan this QR and have a look and have a bit of a virtual experience of how it is to work with it.
Wonderful. Thank you again, Dr. Lodato and Dr. Neumann, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. I would like to note that the Biomech Ingenious will be in Pillar's booth at AMP. That booth number is going to be 619, so stop by if you're going to be there and check it out in person. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. I want to remind everyone that there will be a survey that pops up at the end of this webinar. If you could please take a moment to submit your responses before you leave. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.